The effects of sex hormones on the characteristics and behavior of chickens. There are two main categories of sex hormones: the male sex hormones and the female sex hormones. In this experiment, we use testosterone, which is a male hormone, and estrogen, which is a female hormone. Testosterone is produced by Leydig cells, also known as the interstitial cells of Leydig. Testosterone causes the expression of secondary sex characteristics in male animals. In humans, testosterone encourages muscle growth, deepening of the voice, and the growth of facial hair, for example. Estrogen is produced in follicles in the ovaries. This hormone maintains the appearance of secondary sex characteristics in female animals. In humans, estrogen causes enlargement of the breasts, a high-pitched voice, widening of the hips, and increased body fat. In this study, we examined the effect of sex hormones using chickens as our test subject. Because male and female characteristics are easy to distinguish. We use juvenile chickens because the reproductive systems are not fully developed. This makes it easy to see the changes in secondary sex characteristics after hormone injection. Our objective is to examine the effect of testosterone and estrogen on the secondary sex characteristics and behaviors of chicks. The chemicals used in this experiment consist of testosterone, estrogen, olive oil, and 70% ethyl alcohol. For hormone application, we need 3 sets of 1 milliliter syringes with needles. We also need equipment for raising chickens, including chicken cages with lamps, chicken feed, feed trays, and water dispensers. For data collection, we use a scale, a ruler, and 3 color markers: red, blue, and black. We use 1 to 3 day old white juvenile chickens of unknown genders. We weigh the chicks and then divide them into 4 groups with 6 individuals in each group. For the control group, Group 1, the chicks do not get any injection and the chicks are not marked. The chicks in Group 2 are injected with 0.4 milliliter of olive oil and are marked with blue color. The chicks in Group 3 are injected with 3 milligrams of testosterone enanthate mixed with olive oil. The ratio of testosterone and olive oil is 25 mg per 0.4 milliliter. The chicks in the last group, Group 4, are injected with 3 milligrams of estradiol violylate. To inject the hormones, we hold the chick in our left hand with the chick's head facing our thumb. We keep the chick's body on its side and use our thumb to lift up the right wing. We use our index and middle fingers to secure the chick's ankles. Then we apply 70% alcohol to sanitize the skin underneath the wing. We inject the appropriate treatment according to the experimental design. We must be careful to to inject the treatment slowly. Then we massage the skin where the treatment was applied to help the distribution of the substance inside the chicks. The injection day is counted as day 1 of the experiment. For data collection, we weigh the chicks on the first day and the last day of the experiment. We use a ruler to measure the width, length, and height of the comb on top of the head and calculate the volume of the comb. We measure the dimensions of the comb at the widest, longest, and tallest parts. In addition, we record the color of the comb and the appearance of the wattle and the earlobe. To characterize the chick's behavior, we record whether we see any aggressive behavior and whether the chicks crow, and the day that they start crowing. For this lab, you will enter the mean weight of the chick into the table provided in your report. The weights of each group on the first day are shown here. Please record these weights in the table. The weights on the final day of the experiment are shown here. The length, width, and height of the comb are shown here. Group 1. Group 2. Group 3. Group 4. Please calculate the volumes of the combs and record them in the table. Now, let's compare the characteristics of the comb, wattle, and earlobes of the chicks in each group. Let's compare the feeding behaviors of the chicks in the four groups. In Group 1, Group 2, Group 3, Group 4, Here are the sleeping behaviors of the chicks in Group 1, Group 2, Group 3, Group 4, Only the chicks in Group 3 crow. The chicks start crowing from the third day after testosterone injection. Aggressive behavior is more prominent in the Group 3 chicks than in the chicks from the other groups.